Thank you so much, Fritz, and thank you to the organizers for giving me the opportunity. So I'm just going to get right into it if I can change the slide.
Yeah, that one doesn't advance, it's just a pointer.
Okay, got it. Great. Okay, cool. So just to start from a very, very bird's eye view, I'm sure we can all appreciate that molecular interactions are really central to most biological functions. And we're kind of used to seeing these static images of these interactions, whether it's an enzyme binding a substrate, DNA binding a transcription factor, or a scaffold protein being bound with kinases to induce signaling. But I want to kind of highlight this fact today that molecular interactions can actually be dynamic and or fuzzy. And so here, they actually can lie on the spectrum going from sort of more structured interactions to more dynamic interactions. And just to illustrate this with a few concrete examples out of many really cool examples that are out there. So examples of these dynamic interactions can include static and dynamic components in the same protein interaction. And this is a foreshadowing of what I'm going to be telling you about today. You can have one protein sequence that encodes multiple conformations, and this is an example of p53, which can interact with different partners based on post-translational modifications. And you can have protein-protein interactions that actually embody this cloud-like ensemble of conformations, such as in this example of GCN4 binding to MED15. So the main question that I was interested in for this project was how are these interactions actually encoded in the genome? And the reason that I'm interested in this question is because while we have some really amazing methods right now, like AlphaFold 3, for example, which just came out 2 weeks ago, these sort of methods can't really determine how dynamic or disordered regions bind their partners. And disordered regions actually make up a huge portion of eukaryotic proteomes. So if we don't study these regions, then we're actually missing a big chunk of what's going on in our favorite proteins. And then also, kind of looking to the future, we actually just need more data on how these regions encode function in order to build the predictive models that we have now for example, structured regions. So in order to study these questions, we focused on this model system, which is this PDZ3 domain in a PSC95 protein in human, which binds to this disordered peptide called crypt. And the very cool thing about this interaction is that the crypt ligand is actually binding to PDZ3 at the C-terminus in a structured manner, so it's a static binding mechanism. But it has this dynamic or fuzzy N-terminal tail, which has also been shown to be important for the interaction. Oh, sorry. And this is not just true for this specific PDZ domain and the specific crypt ligand, but actually if you look at the PDB at all PDZ domains that bind short ligands, you see this sort of similar pattern where if you look at the B factor of the crystal structures, you see this increased motion or a higher B factor at the N-terminus, and it decreases as you get closer to the C-terminus. So this kind of implies that this is actually a general thing going on in PDZ domains binding to short ligands. So again, this is a cool system because we have both the structured and the dynamic binding in the same system, so it's well controlled. And the structured component is actually very heavily studied, so we have a lot of data to compare ours to. But the dynamic component is much less well studied, so it's very interesting. And also the PDZ domains it's the largest family of domains in the human proteome, binding to many, many short linear motifs. And so it's just very interesting in general to study how, for example, specificity is achieved in these large protein domain families. So the main questions that we're asking with our specific system basically is how is affinity and specificity encoded in these domains and motifs? And also, does the fuzziness in these ligands actually contribute to affinity or specificity, and what's the mechanism? So in order to do this, we use deep mutational scanning with a very well-known binding PCA assay as the selection. So we basically transform our library of variants into yeast, and we subject them to this selection where the proteins that we are assaying the binding for result in yeast growth under this drug only when they're interacting with each other. So basically, the first thing that we did was just to understand the genetic encoding of the affinity of this interaction. So we made two different libraries that are combinatorially complete, one in the dynamic region and one in the structured region. And we had for each library about 100,000 variants, encoding almost the entire combinatorial space of mutations that are possible. And what we found is that actually both of these regions seem to be quite important for binding affinity, although the N-terminal dynamic region is much more robust to mutation. And when we look at the top binders, what we find is, thankfully, we see this very well-described class 1 PDZ binding motif that comes out of the C-terminal part. But what's really interesting is that actually you also see a very striking amino acid preference that's also position-based in the dynamic part. So you can see that the dynamic part prefers these positively charged residues as well as aromatics. And looking closer, we actually see that net charge is actually super important for binding in the dynamic region of this ligand, but it is not the case at all for the structured region. So this is, again, illustrating that it's actually very useful to be studying these things side by side because you have this sort of built-in control in the structured region. So just to summarize, both dynamic and non-dynamic components of the crypt are encoding affinity. The structured portion is much more sensitive to mutation, whereas the dynamic part is much more robust, which is quite expected based on what we know about disordered regions. And the dynamic portion seems to contribute to affinity via net charge. So what about specificity? So to look at specificity in the interaction, we made all possible double mutants between the ligand and the PDZ domain. So we were able to get around 200,000 measurements of how the peptide is interacting with the domain. And just as a sanity check, we can see that the single mutant data that comes out of the scan recapitulates what we know about the most severe effects to binding being at the binding interface. And in the ligand, the most severe effects are in these two constrained residues in the structured part of the motif. So how did we actually identify the specificity changing mutations? Well, we used this two-state thermodynamic model that we implemented using Mochi, which is a tool that was developed in our lab and is available if you want to learn more about it. We got a very good fit of the model. And if we look at these positive residuals here, we can actually identify the mutations that are binding better than we expect based on this model. And so what we call a specificity changing mutation is basically a variant that has a positive residual that is significantly different from 0, and where the variant is actually binding better than our threshold for a non-binding variant. So it's actually alive. And when we do this across all positions in PDZ, with all positions in crypt, we can actually look for enrichments of specificity changing mutations in each pair of sites. So here I'm just plotting the effect size, which is the number of specificity changing mutations. And we have some statistics about whether or not that's a significant enrichment. And what we see is that there are actually 20 major pairs of sites that are encoding specificity in this interaction. And what's really cool is we can then go into the structure and actually plot for each crypt residue what are the specificity changing positions in PDZ. And we can do this again across the interaction. And what's really interesting about these pairs of sites is actually that they are largely encoding specificity at one particular crypt residue. So it's a very modular interaction. And of course, we have resolution into the specific mutations that are coupled in this interaction. And just to show you that we get this sort of smoking gun mutation pair, which was really beautifully characterized in this study. But beyond this, this very famous class-switching mutation, we see many other mutations that achieve the same effect. And of course, we get the full map of mutations that are coupled and changing specificity. We also have the raw binding scores, as well as the change in binding compared to wild type. And again, we have this for every single position in crypt with every position in PDZ. And I will not go into the summary here for time purposes. But basically, the last thing we wanted to know, because what we found in this part of the study was basically that the specificity encoding is through the domain is mostly through the structured part of crypt. So we wondered if the fuzzy part has anything to do with specificity. And for this, we made all possible double mutants across the dynamic and structured parts of crypt. We again did the same modeling approach, and we found one significant pair of residues, this glutamine here and this tyrosine. And when we plot it on the structure, we see that these could potentially have side chain interactions. And they both interact with the same residue in PDZ. And there are very specific mutations that drive the specificity encoding. And what's really cool is that the side chain in this dynamic part is not actually resolved. So we're able to understand using DMS something that we would not be able to see in the structure. I will skip through this. I will also skip through the conclusions. And yeah, so basically just looking forward, we want to probe the extent of how programmable the specificity is, apply this method to other peptide recognition domains and other molecular interactions. And we also need more DMS assays in general to understand assorted regions. So with that, I'll thank Ben and the lab and funding. And also, we have a preprint that was up a couple weeks ago. Thanks.
Super cool. We have a question here. Very beautiful talk. I really love the combinatorial mutations and all of that. I guess you pitched this as like trying to study the fuzzy interactions, but at the end it seems like mostly the structured parts that are interacting, and of course that's just how the cookie crumbles sometimes. But I guess I'd be curious to hear how you're thinking about studying, trying to go after fuzzy interactions. Like are there other model systems that you maybe think are better suited, because peptides are kind of probably going to end up being enriched for these structured interactions. I'd just be curious. Yeah.
Yeah, I think in terms of the specificity, at least through the domain, it seems to be definitely much more enriched in interactions with the structured part. But the cool thing is that we can also pick up a few interactions that really do seem real in the dynamic part, which you wouldn't be able to do just without doing these experiments. So I think the effect size in the dynamic part is less, at least within the interactions with the domain. But it's cool that you can actually pick up the interactions. And then, of course, at the end, it was a bit rushed, but it seems like the specificity encoding is actually possible through interactions between the fuzzy and non-fuzzy parts of the ligand. So it might not be with the domain as significant, but it is within the ligand.
maybe I can ask, oh, you got another one. Okay, good. Who's on first? I think, why don't we look at you?
Okay, I'm sorry.
No worries.
Hi, Taraneh. That was a really cool talk. I've been thinking since Jean Yue's talk yesterday about this blossom as kind of like a null model, and like wondering, okay, what can DMS show us about sites that have a different tolerance pattern, something that's more idiosyncratic? Have you seen in unstructured regions any tolerance profiles that seem to be just like substantially different from what you might see in like a normal protein region, or just that like stand out to you just based on like the tolerance profile as being unusual or interesting?
Do you mean sorry, but what do you mean by the null model of tolerance?
Oh, so like, let's say you have a site that doesn't seem to be defined by like its charge or hydrophobicity or size or anything like that, and it just is like, okay, this region's doing something different from that. Like, have you found that in unstructured regions, or, yeah?
Yeah, I guess like when we try to make sense of what's going on in these regions, and we basically have like physicochemical features, and maybe there's a motif that might be interesting. Yeah, so I think beyond that, it's kind of hard to think of other things that might be going on. But it does seem to be like a lot of the things that I'm seeing, at least from this data, is like interactions between physicochemically plausible scenarios. So you have like positively charged things interacting with negatively charged things, or you have hydrophobic interactions. Yeah.
Thanks.
Hi, this is Sam Barry from Harvard. Thanks so much for the great talk. I was curious because I know that we know a lot about the natural, like the human PZ domain specificities. Have you looked if any of the specificity changing mutations you found correlate with what we see for there's like PZ domains with different specificity?
Yeah. So yeah, there have been a lot of really cool studies about PDZ domain specificity, and what we find is that our results do overlap with those results, but basically because those results, a lot of them, the sort of mutations that were made and so on, were narrowed down based on the evolutionary data. But since we kind of take an unbiased approach and just mutate all of the sites, we find kind of significant interactions beyond what was found before, but a large overlap as well.
But like, are there any like of like the other human ones? Do you see like, oh, we found that like a G to Q mutation here has some effect, and we also see that in this natural human protein.
I see. Yeah. So I mean, for example, there's like class 2 PDZ specificities. There's different classes that they have been classified into, and we do see all of those across in our data. Yeah.
Thank you.
Okay, great. I see you've got more questions, but maybe they can find you after the session,
Yes.
and we'll keep going. Thank you very much.